Hey, this is Joel Duff. Welcome back to my channel. And today I asked the question, did Adam and Eve have tails? Yeah, I know. That's a crazy question. Of course Adam and Eve didn't have tails. Or if you prefer, did the original human beings have tails? Whatever you think about the origins of human beings, did they have tails? For Ken Ham, he answered a couple years ago when this question came up first. Of course, humans don't have tails. But I would ask Ken Ham, were you there? Did you see Adam and Eve? Is there a record in scriptures in the Bible that gives us a description of Adam and Eve that tells us that they lacked tails? After all, when Adam and Eve looked around at all the other animals, especially all the other mammals and, well, actually all the mammals and all the dinosaurs, he would have seen a lot of tails. And they looked at each other. Did they see tails or not? That is the question, right? But the way we're going to ask this question is we're going to look at the molecular genetics, the genome information that allows organisms to make a tail or not make a tail. And what's the difference at the genetic level between making a tail versus not making a tail? So this is going to be a geeky molecular genetic discussion. And it's one I've had before. So if you're a long-term follower of mine, you know that I've already used this title and I've already done a talk on did Adam and Eve have tails? And I went over a paper in excruciating detail. We're going to go over that paper again because you know what? That paper has finally been published. 2 years ago, I talked about a preprint, right? Something that was not yet reviewed and had not yet been officially published. Well, 2 years later, the paper has finally been published in the journal Nature, one of the top science journals. Turns out they wanted some extra work done to bolster their evidence, right? Provide us more evidence for the claims that you're making. After all, you're making a huge claim. You have found the mutation that has caused the loss of tails in primates. Well, apes, great apes anyway, of which human beings, don't have tails either, right? And so they requested that, and the authors did some really cool experiments, which I wasn't able to talk about before because they weren't in the paper before, and now they are. And so I'm re-upping this one with this new information. And I've had time to think about the implications of this research to the whole question of origins and how do we know what we know? Did Adam and Eve have tails? How does Ken Ham know they didn't? And even if Ken Ham doesn't know, how might we know whether Adam and Eve or the original human beings, whoever they were, did or didn't have tails as adults? I got to toss on that very important caveat, as adults, which you'll see why in just a few minutes. Okay, just as a quick reminder, this was Ken Ham 2 years ago, which is the whole reason I even did a video in the past about this. He had reported on what happens once in a while, which is some baby is born with something that seemed like an extension of vertebra. And so it appears that they have a tail. And I talked in that video about what is that truly the same type of tail as other animals have tails? And it's not exactly the same way. It really is a mutation, a developmental error that creates the thing that looks like a tail. And so Ken Ham's not wrong, really, that these aren't really the same kind of tail as the type of tail that other primates and other mammals have. That did get me looking into how is it that we don't form a tail? What is the genetics of that? And there was a paper that had just come out in preprint server, which I mentioned before, is just that's where they're hey, this is a manuscript we have, and we're going to get feedback on it before we actually submit it for publication. And so I saw that manuscript, really interesting research that they did, talked about it, and that's what I'm going to talk about again here. But before we get to the paper, let's set the scene with some questions that probably very few other people are probably asking about that paper, right? So if you're the authors of that paper and you notice that I'm going to review your paper and talk about it, these are probably not the questions you had in your mind when you wrote the paper. These aren't questions you were trying to answer. But here's some observations. And actually, these would be observations that they made. And one of the reasons why the primary author of the paper, was he actually talks about how he was pondering the whole question of like, why don't we have tails when like all other vertebrates, land vertebrates do, right? So we observed that all vertebrate animals have tails at some point during their life, right? And virtually all vertebrate animals have tails as adults. There are a few exceptions. Like I say here, there's some frogs that don't have tails. There's some bat species, and there's a couple primates, right? Gibbons, chimpanzees, bonobos, gorillas, and human beings. And when I say that all vertebrates have tails at some point, what I'm referring to is embryologically, all right, developmentally. All right, in the embryonic stage, every mammal that has been, or every vertebrate that has been studied from an embryological point of view, begins with tails and has the beginnings of the makings of tail vertebra. Often they're just connective tissue, right? They haven't turned into bony tails, but nonetheless, they have the beginnings of the vertebra to make an extension of their spinal column as this extension of their body, right? As a tail. And so human beings and turtles and fish, right? Chickens, go on down the list, whatever animal you want to think of, and you look at embryologically, they have a tail. And so you start out with a tail, but then some organisms lose that tail prior to birthing, right? Prior to becoming, well, let's just say adults, right? They don't have a tail as an adult. So that's got to be a developmental thing. There has to be the genetic capacity to make a tail, but then there has to be something that's telling the organism stop making the tail. In fact, not only stop making the tail, but reabsorb the tail, like actually break down the tail that you've just made, reabsorb it and turn it into something else. So that brings us to our provocative question. Why don't humans then have tails as adults if most other animals have tails? I can think of three hypotheses. And really, I'm addressing here primarily Christians who would be interested in the question of what would be the original state of mankind. The original state, like say like Adam and Eve as the primordial foundings of the human race. Did they have tails? Right? God made Adam and Eve without tails as adults from the beginning. That's one proposition you could have. I'm sure this is what Ken Ham would say. There's Adam and Eve. I mean, he's got a creation museum and he has Adam and Eve there, and he has a depiction of what they look like, and they don't have tails. I've been there. I've seen it. I didn't see any tails. Right? Their experts there have decided that Adam and Eve didn't have tails. They weren't there. They didn't see Adam and Eve. Again, there's no written record of them having a tail or not having a tail, but they depict them as not having a tail. And I'll talk a little bit later, if I remember, about why there's good reasons to believe that, aside from the scientific evidence I'm going to show you. Another hypothesis would God made Adam and Eve with tails as adults. Adam had a tail, just like he had a belly button, even if he was created de novo. But then they were lost in their descendants via, and I think this is how young earth creationists would maybe explain it, as by mutations. Natural selection, genetic drift. The processes of change have reduced or lost those features. After all, young earth creationists love to talk about degeneration, right? They love to talk about genetic entropy. Characters are being lost. Organisms are being simplified. And so just a genetic switch change could have caused them to lose their tail, but they still survived, right? So we're depauperate. We don't have what we once had. In the perfect creation, we had tails. We could have climbed up into the fruit trees and gotten the fruit using our tails to our advantage, but we don't have those anymore. That's made life more difficult. I mean, Adam sinned, and as a result, a mutation happened, and poof, our tails are gone. Now that would raise, if that's a real possibility, and I don't think anyone can exclude that possibility, at least from this young earth creationist perspective. And then the third option would be adult tails have never been expressed in any person. And the reason for that is that the physical bodies of human ancestors, in other words, ancestors before Adam and Eve, or before first individuals we called humans, once had tails, But then they were lost via mutation and natural selection. In other words, the first individuals we'd call humans, so like in an evolutionary creation or theistic evolution perspective, if there are prior generations of ancestors of humans, maybe they didn't have tails, but maybe their ancestors had tails, right? And eventually Adam and Eve didn't have tails because they're descendants from individuals that lost their tails. All right, sorry, that was a long way around saying something that I should have said much more simply. Okay, now with that prelude, with those three separate hypotheses, let's take on the challenge of getting to the super geeky molecular genetic stuff and look at the actual genetics of tail formation or lack of tail formation. And here's the paper we're going to take a look at on the genetic basis of tail loss evolution in humans and apes. All right, on the genetic basis, so these authors, this large group of authors, because this is a lot of complicated experiments that I'm not going to try to explain all of them. I'm just going to give you a taste of some of the interesting details here. They claim that they have found the genetic basis for why some primates don't have tails, right? And their explanation is that they did have tails, but they've had a mutation, and they think they have found the mutation. They think they have found the change that has occurred to the genome that has resulted in losing tails. So Bo Xi and colleagues from a variety of different institutions, all collaborating on this very complicated project, but it really comes down to a very simple piece of data. And this is why I like this particular article, because although there's a ton of complexity to it, it's actually fairly simple to see what happened here. And that's what makes such an elegant science story, is because it could have been a really complex story, but in fact, it's actually kind of a simple thing that potentially happened in history that caused the loss of the ability to make tails in primates. So before I get to a couple figures, because I'll just mostly stick to the figures, let me tell you about the gene we're going to be talking about. They scanned hundreds of genes that are known to be involved in early development and development of spinal column and bones and all the things you would think would need to go into making a tail, right? We have a pretty good idea of the different kinds of genes that are involved in that process by studying all kinds of different organisms that have tails, right? Especially mice. Right, so we narrowed it down to here's a suite of 100 different genes that could be involved. And they then started to look across all those different genes and they asked the basic question, what's different about organisms that express tails as adults versus ones that don't express tails as adults? Are there genes that are missing? Like maybe one of the explanations is one of the genes is gone, and that was an important gene for making a tail, and because it's not there, you can't make a tail. Is there like an addition of some new gene that all the other ones don't have that somehow prevents the formation of a tail? Right? These are all the types of things you'd first wonder if you started a project, just asking what is the difference between an organism that makes a tail and an organism that doesn't make a tail? When obviously they can make a tail because they make them in the embryological stage. And they narrowed it down to, and I'll show you why in a moment, one particular gene they think is the most important in terms of starting the process of tail loss, TBXT. It's an early development gene. That just means it's a gene that comes on and is active very early in the developmental stages of life, of a multicellular vertebrate. The TBXT gene, which is also known as T brachycura gene, or longer name for it, T-box transcription factor T, okay, is found in bilateral animals. All right, bilateral animals, that's an awful lot of animals, ones that have bilateral symmetry. So it is a very old gene, or a gene that is found in lots of different organisms, a very conserved gene. It's a very important gene for very essential early on development. Plays a critical role in human development as well. It's a member of the T-box gene family, which is involved in the regulation of developmental processes. It's a regulatory gene. It means it's a gene that makes a product. That protein product then, depending on when it's made, where it's made, and how much is made, will influence what other genes are turned off and on. And that's what causes cells to differentiate and become different things inside of a multicellular organism. Stimulates them to grow, stimulates them not to grow, stimulates them to change into neurons versus bone cells, right? So those type of regulatory genes are the ones that are really, calling the shots. So it's essential for the proper formation of the notochord and the development of the mesoderm, which is a key embryonic layer, which eventually gives rise to muscles, bones, and other connective tissues, right? So it's so early. It's the thing that's like tripping the whole set of other things that will come on and say like, okay, here's a bunch of cells that aren't really differentiated. Now, when this gene comes on, it's going to influence all these other genes to then cause these cells to then differentiate into all kinds of other things. So now let's look at that gene. So this comes from figure 1 from the paper. And I labeled this as comparative sequence shows something different is found in great apes. They did a really simple thing. They looked at sequence comparisons of genes that are like important developmental genes. Ones that we know we can see are turned on and have it in other animals like mice. We know that this gene is turned on at a certain time, and when it's on, the development of a tail happens. And so they just compared sequences. And they said, is there a difference between organisms that are making a tail and not making a tail just in the sequence itself? And if there is, maybe we'll then know maybe we can look at that particular sequence and then determine whether that change in the sequence is actually the cause of the change in development. And so what I'm showing you here is, let me show you all the stuff that's on this. At the top here, we have human chromosome number 6. This is the entire human chromosome number 6. It's like, 75 to 100 million base pairs long. And then this little red bar all the way down here represents where the TBXT gene is on chromosome number 6, right? There's hundreds of other genes. They're just looking at this one gene. And then they expand it out. And then so like base pair 160, well, let's see, what is this? You got 3 zeros here. Yeah. 166,166,700, right, is right here. And then it goes down to 166,159,000. So this is, about 8,000 base pairs of ATCs and Gs. 8,000 base pairs of sequence of the millions of base pairs in that particular chromosome. So this is the location, the locus of the gene in genetic language. And in that gene, we then have, you see these colored boxes? Each of these colored boxes represent exons. There's 8 exons. Exons are expressed. Those are maybe a simpler word for it. Those are the coding sequences. Those are the sequences that have ATs and Cs and Gs that code for making a protein. And the protein is the regulatory protein that does the thing inside the cells, right? In the tissues. And so you have to put all these little exons together. I have another figure that's going to show this. You put the exons together, and then you read the language of the DNA, which is now an RNA code, and you turn it into this protein. What's in between is called introns. Introns, intervening sequence, because they're intervening in the coding sequence. And the intervening sequences have to be cut out prior to reading the code. Again, I have a figure, but we'll go over this again. But for now, that's all you need to know in order to look at this figure. Other than the black line, right? What's the black line? Chimpanzee. Here's a long black line. Black represents the same sequence as human beings. All right, so this is ATCs and Gs all across here. I'm not showing them because you can't compact 8,000 base pairs there. We're not going to spread this out that far. And so this represents chimpanzees. If it's all black, if it was 100% black across there with no white lines, that would mean it would be 100% the same. Chimpanzees have the exact same 8,000 base pairs. And you can see it's pretty close. There's a couple little gray spots here where there's like 1 base pair different. And then there's one little white area here that's probably like 10 base pairs that is missing, all right, in the chimpanzee genome that human beings have. So there is some genetic difference between chimps and humans. And then you have bonobos, which are basically identical to the chimps. And then you have gorillas, and they're pretty similar too, although they're not actually lacking this little deletion. They have the same sequence as human beings. orangutan, and so forth. And then you notice down here we've got a bunch of other types of monkeys, marmosets, and all that. So you notice one big difference? Here's the big difference. There's a big chunk missing right here. That's more than 300 base pairs. Right, that's a little more than 300 base pairs that is missing in all of these monkeys. Well, what do all these monkeys have in common? From the black snub-nosed monkey all the way down to the white-faced savajou, they all have tails. They all have tails. What do all the ones that have sequence there have in common? They all lack tails. All right, now that in itself, it's an interesting correlation, right? That's an interesting observation. Those are the facts. Those are the facts of the genome. These sequences, these genomes have all been sequenced from all these different species, and all the genomes of organisms that have tails have 300 extra base pairs here. So now, of course, what they say is like, okay, well, that's a difference. We're not seeing that in like the other 50 genes that we looked at, where we're trying to look for other genes that could be involved in tail formation. It's like one base pair here, two base, it's like there's no obvious correlation between organisms that lack tails and ones that have tails. This is like the first one that jumped off the page at him is like, wow, there's like a real difference between these two. What could it mean? Well, let's look at what the sequence was. You'll see there's something else in this little cartoon up here. We have our colorful boxes, I said are exons, but you notice that these two boxes right here, this gray box and this red box, do not have the word exon next to them because they are not exons. They're not expressed sequence. They do not make up part of the code that makes the protein. They're part of the introns. And these two have names down here. You see ALU SX1, and then we've got ALUY. And then they've labeled this hominoid specific because these are all considered hominoids in terms of their classification. And this red box then represents a sequence which is specific to just to the hominoids. Now, the reason they have names is because these are known types of elements that are in genomes, the ALU element. And the ALU element is a generally 300 base pair jumping gene, all right? It is a transposable element. It is a piece of sequence that has the capacity to copy itself and insert its own sequence back into another place in the genome. And this is how it moves around. All right, what is the significance of this little ALU transposable element? Well, TBXT gene, it has a different structure because of this extra 300 base pairs. Now, I could if I just threw in 300 extra base pairs into an intron, it most likely wouldn't make much of a difference. The gene would probably still read the same way, and it might get copied the same way, regulate the same way. But this 300 base pairs is a little different, and it has caused a specific thing to happen. And this is what's really wild. All right, this is really cool stuff. Here's what happens if you don't have that extra ALU unit, that extra 300 base pairs, right? You've got a gene and you've got exon 6 and exon 7, which are on either side of this new little ALUY that's inserted. Let's say you don't have that insertion. That's what this is showing here. Saying like, here's the gray ALU SX1. That's an ALU identified unit in the genome right there. But on the other side of exon 6, the other one isn't there. That's like the tailed primates, right? If that happens, then what happens is you need to cut out this intron and you need to cut out this intron and you need to splice it all together. And when you splice it together, you then create all 8 exons that you then copy into a protein, and it does what it does. It regulates, and what it does is it leads to making a tail. Now over here you got ALUY. So the other option would be, instead of having exon 6 with this ALU unit on both sides, sorry, on only one side, you now have an extra ALU unit on the other side. And the reason the arrow is pointing in the direction is because it's saying like the insertion of this 300 base pairs is actually reverse complement. It's the opposite direction, pointing the opposite direction in terms of its sequence as the other one on the other side. And it turns out that when you make your messenger RNA, and I'm going to, again, I am going to talk about all this twice. All right. And so I have two chances to try to explain this. This is my first just graphical explanation. Here's the exon 6. And because there are two AL units and they have opposite sequences pointing in the direction, when the messenger RNA, the message that you've copied from your genome information, it's single-stranded and it will fold up, and the two sequences will find their opposite sequences because they're almost the same sequence. And so they will line up and they will bond to each other. Just like, you know, the ladders of the DNA ladder, the RNAs will bond to each other right there, forming a loop. And when that loop forms, it then cuts out this. Let me get my pen. That might actually help if I can draw. That's going to end up cutting out this section. And then this exon and this exon will be grouped together, missing exon 6. So if you have the extra ALU unit, it creates a mutation by which you now start to chop out the exon 6. You splice it out. When before it was still there and it was part of the expressed reading sequence. Now you're cutting it out. And when you cut it out, it turns out lacking exon 6 makes a different protein. That different protein behaves differently. It regulates differently. And as a result, no tail. Now that's, I'm scared to say that's the simple explanation. I will say it is, but I'm going to explain it again. Let's go back through some of these elements again. Elements, the ALU element. What is that, you're wondering? ALU elements are a type of transposable element. They can transpose, move themselves. Found in the primates, the genomes of primates. This specific 300 base pair sequence, there's variation in 300 base pairs, but it's kind of has a similar profile, similar genetic structure in terms of the code that's there. It's found in primates. Right, so it's a sort of a primate-specific repetitive code that can move around and transpose itself, including human beings. They're part of what are broadly called short interspersed nuclear elements, or signs. Those are things that, like, all eukaryotes have, things that have a nuclei. And they're present, and they're the most abundant transposable element in the human genome. We have lots of repetitive sequences in us, in our genomes, all of us do. But this particular version, this particular class, accounts for 10% of your genome. 10%. Right? So if one copy of your genome is some 300 million base pairs, 3 billion base pairs long, 300 million base pairs of your genome are repeats of this 300 base pair sequence. So imagine how many copies of that 300 base pairs you have. They're everywhere, scattered all over in your genome. All right, and so I think I already mentioned the most important. This is a slide from my class where I'm talking about something else in genomics. 300 base pairs long. They have, let's skip this. We don't need it. You don't need to know this. But you do need to know this about TBXT. The gene is located, as I already said, on chromosome number 6. It has several exons. And those exons undergo different kinds of regulation by alternative splicing. Alternative splicing is when you mix your different exons together to make different proteins. So that was already known. We already could do that, right? How does that work? Let me explain that, because that's the really critical part of understanding this particular interesting mutation. So let's just look at a typical gene in human beings, really any mammal. A single gene might have, you have something that tells it, like, here's how to turn it on, here's how to promote it, like, I want to use this thing. So there's some code or sequence here. And then you have a series of exons. exons are express sequence. Like, okay, you're going to start reading your code here, and then you're going to read it through here, and then you need to read this code, and then you're going to read this code, and you're going to read this code, and all of that makes a protein. And then you have some other place that, some other sequence at the end that helps you transport that particular sequence. Well, what happens is you have this code, including the introns, right? Intervening sequences. All of these are in your genome. So in chromosome number 6, there's that location. It was like, oh, early on in development, 2 days, cells divided into 2 or 3 cells. And all of a sudden it's like, I got to turn this gene on because we need to start thinking about how we're going to do development, right? So to turn the gene on, what does that mean? That means you go in, you find this sequence for this gene, TBXT, and you copy this entire sequence out, right? Copy all this stuff out. And when you have it copied, you can't just directly like read that sequence and translate it into a protein. You first have to remove the introns. So you remove this portion, you remove this portion, remove this portion. You put the 4 pieces together and you end up with a messenger RNA, the message from your nucleus. The nucleus stores your information, right? Your code, and then you send the message out to the rest of the cell where it can be read so that you can use that information in the form of a protein. So ultimately, you're putting those 4 exons together to make one single reading transcript, and then you're going to read it into a protein. All right, but then you got this thing called exon skipping technology, or just exon skipping, or exon shuffling sometimes. And so you've got your 4 different exons, and those are the 4 parts of your gene, your code. And then normally what you do is you cut out the introns after you've copied your DNA into the messenger RNA, the message that's going to go out of the cell. And then you would splice them together to make the final sequence that you're going to read. But what if you have a mutation in one of the exons such that this particular exon doesn't get spliced out? Let's say it just splices this whole thing out, right? And you splice out the exon too. Well, then you're going to put these 3 pieces together. And when you put these 3 pieces together, you're going to make a different protein. Now, often that protein might not make any sense. It doesn't have any job to do. But we have many genes in which we have 20 exons. And 15 of the exons make this protein. But if you add 3 more exons onto it, you can make a different protein which does a different responsibility. This is how we can have 23,000 genes, but we can make 100,000 different products. You realize that? We make like 100,000 different proteins, but we only have 23,000 genes, right? Or protein coding genes. Because some of our genes are doing double duty, making multiple different products. All right, so now what can happen is, how do those different products get made? Well, they get made by alternative splicing. I could splice all of this out. I could splice these 2 sections out and only keep these 2, in which case I'm only going to have a protein that is made up of the information from those 2 exons. What does that have to do with this? All right, all of these other primates and all other mammals that have tails, which is almost all of them. All right, they all have the TBXT gene. And guess what? Their TBXT gene looks just a lot like ours. It's a whole bunch of exons. They have to splice them. They have to put them together. And they do some alternative splicing where they use different exons in different ways. That's how it's able to do development in different parts of the body differently. But all the ones that have tails are lacking this particular ALU unit. And as a result, they do not splice out exon 6. Exon 6 is always part of the final product, right? The information from exon 6 ends up being in the protein. But those organisms that have this new 300 base pair sequence that nobody else has, they use that to cut out this entire segment. Rather than just splicing this segment and this segment separately, the 2 cause really a mutation, creating a new splice that splices out that exon, putting together all the other exons together and creating a different protein a protein that typically isn't produced in any of the other animals that have tails. And somehow that interferes with the developmental process, causing the tail not to form, and then it gets reabsorbed by the body. So back to this other figure again. This is the same figure we saw before, but now maybe this will make a little more sense here. Where's my line? Oh, there it is. Here we have ALU, and it's saying like, here you got the ALU unit, here's the ALU. How does it splice out? Because it forms this loop, the splicing mechanism here and here will end up splicing this, which ends up splicing the whole thing out. And so as a result, in human beings, we make a version, as well as some of the other, as the other great apes, we make a protein version, regulatory protein version that is missing exon 6. By the way, we do make a full-length exon 6 too, because this doesn't happen with 100% efficiency. So we still do make some copies of our proteins that have exon 6, and we have some copies that don't have exon 6, a lot of copies that don't have exon 6. And apparently, having that imbalance creates a problem with tail development, right? If we had two copies always making TBXT exon 6, presumably we'd have tails. All right. Now that's a lot of like, I'm saying, oh, well, because we have this extra 300 base pairs, we don't have tails. Right, how do you actually show that? Like, is there a way to like, is that just guessing? It's like, isn't that just a correlation? I thought correlation didn't mean causation. All right. Here's where the extra couple years of more stuff that these people did before they published. All right, the first thing you need to know, the first step to showing causation would be to say like, all right, we're predicting that having this extra 300 base pairs is going to create a fold in the messenger RNA that then will cause it to be spliced out, and so exon 6 won't get incorporated into the protein. Can we show that? Well, one way they showed that was, I'm going to skip forward here. One way they showed that was to show that in human beings, they actually get the protein itself, and they show that human beings actually make a protein that is missing exon 6. The information for exon 6 is not there, right? It's lacking. So we really don't use exon 6, even though we have exon 6 in our DNA. Right? So we make a protein that is missing exon 6. So we know that it skips that particular exon. Then the next question is, how do we know it's skipping it because of the extra 300 base pairs? That's where it gets kind of neat. They went in and they did genetic engineering on cells. So they take a cell line of human cells. They take that cell line and they use genetic tools, CRISPR tools, in order to cut out the 300 base pairs, right? They're indicating like, hey, what if we cut out this thing right here? And then the other thing we can do is we could cut out ALU SX1. We could make two types of cells, and we could have some cells that are missing exon, the ALUY, and some cells that are missing ALU SX1, and we could see what happens. And then you grow those cells and you ask the question, do those cells make proteins that include exon 6? And guess what? If you cut either one out, they do make proteins with exon 6. In other words, we have the capacity to make full-length proteins, including exon 6, in all of us. But in many cells, we're not doing it because we have this extra 300 base pairs, which is interfering with that and shutting off the developmental process of making a tail. So that's one experiment. That's done in human cells to show that they actually express the protein, and they can't cut out exon 6 unless they have both ALUs, one on each side of exon 6. So that shows that what they predicted based on just what we know about molecular genetics and doing all kinds of modeling and stuff, what we predicted actually happens in cells. They just they skip that exon. What was more interesting to me was the fact that they took this and they said, let's do this in a mouse model as well. So mice have tails, right? Why do they have tails? Well, they have TBXT along with lots of other genes, and they have an exon 6, and they use exon 6 in their protein. They said, well, we can use our genetic tools and we can just cut out exon 6. We'll just like cut it out of the DNA so that it doesn't even exist. So when they go to make the gene, right, make the product, it'll be missing exon 6, kind of just like a human being, cutting it out, and so not including it in their protein. So they did that, and sure enough, you cut it out and you create a mouse that makes some copies that are missing exon 6, and they have short tails or no tail at all, or some other type of genetic malformation in their spinal cord. All right, so let's look at the big picture here. What are the authors suggesting actually happened? What they're suggesting is that there was a time, sometime in the past, you have great apes, all of which lack tails, and they all have the same exact sequence that's inserted into their genome compared to all others that have tails. And so the simplest explanation for that would be that there was a point in time where there was an ancestor of great apes. That ancestor of great apes would have had a tail because they're a mammal and mammals have tails. And then they had an insertion of the ALY insertion. Not an insertion of the other ALU, that was already in all other primates already, but apparently didn't really do anything, right? Doesn't seem to have an actual function. It's just sitting there. It's like it got inserted. You have millions of ALU units, and the vast majority of them are just like plopped in at various places, and they're not doing anything. You can think of it as just random stuff hanging out in your genome. But then this other ALU unit just plopped in, and I guess by chance, right, happened to be on the other side of the exon 6, situated in the opposite, reading in the opposite direction. And as a result, it created a fold that didn't exist before. The messenger RNA will fold up and it will align with itself because it has the opposite sequences. And I'll say base pairing sequences. And as a result, it then resulted in the splicing out of exon 6, which caused a developmental change, since it's such an important developmental gene, developmental regulator. So that would have caused, now this sounds really simple the way I just put it, but it wouldn't have been that simple. It's not just like I have a tail, don't have a tail. It's not that simple. Almost nothing, no complex characters is ever going to be that simple, but it is as simple as that was probably the trigger point. Like that was the significant mutation event that caused a significant change in morphology, creating, in this case, you'll see here that they're showing like, hey, the offspring of that monkey that had that mutation had maybe longish tails, but not as long, maybe no tails, stubby tails, maybe varieties of tails, right? And this is what happened with the mice, right? If you take this gene out, this piece of the gene out, it creates mice that have a variety of different forms of malformed tails, all right? It's not that single gene is enough to completely change tail formation, right, into utterly no tails. But then what they're suggesting is this wasn't lethal, all right? Having a little bit shorter tail, something wrong, kink tail or whatever, that wasn't lethal to the individuals that had that mutation. And then they had additional mutations, not necessarily in the TBXT gene. In fact, we don't think that they were in the TBXT gene because you can compare the rest of the sequences. They're really similar. There's not a whole lot of difference in the rest of the gene. However, there are 50 to 100 other genes that are responsible for making tails. Now, once you don't have much of a tail, if you have a mutation in another gene that also is involved in tail formation, like maybe a little later in the developmental process, and it creates a better version of no tail, because quite honestly, if you just have this mutation, we know by looking at the mice that it creates other neurological problems, right? They can survive, but if they have other mutations in future generations, they'll become healthier without that tail. I don't know if that makes sense. So there's Well, all right. Here's how to make sense of this. The authors of this paper have associated. Well, actually, I don't think they had to associate. I think there was already a known association with TBXT and neural problems like spina bifida, right? A neurological malformation, right, in humans, which is a fairly common genetic disorder, right? And that's been traced to genes that are involved in tail production, which are involved in all kinds of other parts of development as well. And so what these authors suggest is that the creation of having lacking tails, the loss of tail production, resulted in an increase in numbers of genetic malformations in neurological stuff. Sorry, not talking very specific scientific language today, am I? Neurological stuff. But the proposal is that lacking tails was not lethal in itself and survived and possibly had advantages, right? Especially advantages for animals that are more likely to be on the ground and forage in different ways, right? Chimps and orangutans and gorillas and human beings all spend more time semi-walking, right? Or for human beings, walking a lot. And tail use is not really as important for them. Well, we don't have tails, but maybe they didn't need tails as much even in their original state. And so natural selection might have selected for any new mutations that made the tailless condition more stable. Right? It might have started out as being like, this isn't really the best trait. It creates other neurological problems. But if we mutate these other genes such that they then sort of mitigate some of the problems, then you end up with a package of genes that then allows you to not make a tail and still be relatively well off. Except, as I said, there's still going to be a certain types of mutations that occur in this particular gene that will result in different genetic conditions that even pose problems today that are more common. We know that they're more common in humans than they are, say, in mice, which have tails. So our lack of tails might be actually resulting in more neurological genetic conditions. All right, so the model is that you have this insertion was like the event that caused an actual morphological physical change, which then natural selection looks at and goes like, is that a good thing or not? Maybe it's actually a good thing for some individuals in this population, and therefore they survive, and then they have more mutations, and then natural selection picks individuals that are better survivors, and therefore sort of takes that and hones it and makes it into like, okay, this no-tail thing can really work, and we'll make it work better over time to the point where most no-tailed primates, the great apes, are reasonably healthy without having tails. All right, so now, all of that backdrop, we come back to yeah, our provocative questions, right? About Adam and Eve. So in a young earth creationist scenario, where you have fiat creation of Adam and Eve, not from any animal ancestry, what you have is you have this gene. Well, let's go back to the gene. I should probably talk about what I have this on. You have this gene, right? And it has this 300 extra base pairs. And you could say like, well, that's the reason why that's common design, I guess, right? They're designed to have this extra sequence that will then cause the removal of exon 6. Except that we still use exon 6. And then all these other great apes also have the same sequence that human beings have. So if you're going to say Adam and Eve are de novo creations, they have to be created with the same sequence that causes a tail not to form. See, what's interesting here is that basically we're built to have tails, right? That's the original design. That's why I asked my question at the beginning. Did Adam and Eve have tails? And were you there? And how do you know that he didn't or they didn't? How do you know? Because when you look at the genome is built to make tails. you have to add this little bit of new information, or you have to add this little piece of sequence in order to stop the tail from forming. And you could see that in the embryos, right? You have the beginning of development, possibly controlled by some other genes and other developmental genes. And then TBXT comes along soon after there and goes like, no, 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 we're not making this tail thing, right? Cut that out. Let's suppress that tail thing. You're not supposed to have a tail. Interesting. Interesting way interesting to think about that. The default is to have a tail. And this is like overriding the default program. So once again, this is just another example of, for a young earth creationist, one of the critiques will always be like, of something like this when somebody else sees data like this. I say like, why would God create something that is like the default is having a tail and then like make no tails? Why not just create a system that doesn't create a tail from the beginning? It's like, why even start to make a tail? You're not going to have a tail. The plan is to not have tails. So why begin to have a tail? And again, I think young earth creationists are going to default back to some sort of like, well, this is just common design. We took the design of other primates and all other mammals. Like, here's the gene, here's a gene that's involved in tail construction, and all other mammals have these 8 exons. So God started with okay, I'll start with the generic genome of a mammal. Well, it's got 8 exons. Wait a second. One of these exons don't really need it all the time, right? We don't need it all the time. So let's put in something there that allows you to like remove it so that we won't make a tail, because otherwise, Adam and Eve is going to have a tail. But that also brings me to my question again, which is, why couldn't Adam and Eve had tails if that was the original design? The original design could have been they had tails, because remember, I hear young earth creationists all the time talk about ALU units and transposable elements, them moving around as mutations that are the result of the fall. Adam sinned, and then all of a sudden little pieces of genes, little pieces of DNA started to move around in the genome, and they started to like bump into things and create problems, right? And change the functions of things. They'll actually show like, oh, here's an ALU unit and it has this particular function, and maybe that function was a post-fall function. So we could flip it on its head. I could say like, maybe Adam and Eve were created with a fully functional version of this gene. And then sin caused a disruption in their genome. And then one of their descendants had this particular gene disrupted, and that resulted in them not having a tail. Right? Adam and Eve had tails. They had kids that had tails. And then some of their kids, 1 or 2 of them, didn't have a tail. And they were like, oh, you don't have a tail. That's kind of cool and interesting. They survived and they had offspring. And eventually, for some reason, the ones that had tails ended up not reproducing. And now we have only no tails. Right? Why couldn't Adam and Eve have had tails? It would have been so simple. Clearly, there's not a whole lot you'd have to change. And you could explain all the reasons why tails don't exist through mutations. Right? Option number 2, I think, for young earth creationists. They could say humans were created with genes capable of inducing tail production, but was designed from the very beginning to be turned off. Yeah, you have the ability to make a tail, but you were created with this option turned off, which raises the intriguing possibility that the option could be turned back on, right? Maybe that program is still there. After all, there are young earth creationists who believe that there is pre-programmed traits, like Institute for Creation Research. Like there's little things that are built into the genome that aren't being used right after creation. And then maybe after the flood, the environment changed, and it's like, I've got this program that I wasn't using before, but now because of this new environment, I'm going to start using it. Right? Maybe after the flood, there was a mutation that was tripped that would turn off the ALUY, transposable element, remove it or have a mutation in it on purpose. And then all of a sudden, we would have had tails again. And maybe that was God's plan, to bring tails back. They were put in the original human condition, but not expressed. But maybe they'll be expressed 1,000 years from now or 10,000 years from now. because maybe we'll need tails in the future. So maybe this is pre-programmed stuff. You see why I don't, I'm sorry. It's just kind of fun to think through these different scenarios, but I have a serious point. All right. And my serious point is that common design is a tricky thing to employ. And young Earth creationists, and I'm really talking about Ken Ham here right now, he's so confident about what he knows. Like he goes to these articles, like Ken Ham goes to these articles and he just immediately like makes fun of like, oh, evolutionary biologists think that, human beings, that seeing somebody born with a little bit of a tail is like, that's like a memory of like that we had tails at one time. And how ridiculous is that? What's ridiculous about it? I just showed you that there's a gene there that you and I have all the genes we need. We have all the code, all the sequence we need to make a tail. We simply have turned off the ability to make a tail. Now, it's not just this gene. I'm going to give that scenario before is that this gene would have been the primary thing that turned off tails. Then there's probably been other mutations in other genes. So it's like a single change here, a little change here, a little change over there, and combined all those make tails unable to express themselves in human beings. And so it's not a matter of like, if I just turn this one thing back on, all of a sudden tails are going to reappear, right? I mean, nobody's going to do that experiment, but I would predict even if you did the experiment where you took out this 300 base pairs, all of a sudden we wouldn't be like suddenly making tails. I think we'd have weird genetic malformations, but we wouldn't have like legit tails. Why? Because there's other genes that were also part of the process of development. Development is a really complex thing. And so once you've changed a few other developmental switches, then it's kind of hard to go backwards, right? Because you'd have to change all those other switches back. And that's not likely to happen. But the point still stands. In the original perfect creation, why not tails? Right? Why not tails? But as soon as you admit that human beings could have had tails, you then kind of have to ask the question, well, that also means chimpanzees had tails, gorillas had tails, orangutans had tails, right? All of them should have also had tails as well. Does that mean they all lost the ability to make tails in the exact same way that human beings did? Because it looks like they have the same mutations we do. That's more easily explained by common ancestry. That's the power of the common ancestry explanation for the lack of tails, right? You could go back to a primate ancestor that had a tail. They then lose the ability to make a tail. Then all their descendants share those mutations, right? And that's why they're all grouped together, because they all have the same sequences as a result. So my challenge to Ken Ham and other young Earth creationists who wish to ponder this idea is Ken Ham seems utterly convinced that Adam and Eve didn't have tails. But I'm saying he doesn't have biblical evidence of that. He has no biblical evidence that Adam and Eve didn't have tails. All right, what biblical evidence is there that Adam and Eve didn't have tails? I mean, I admit it's an easy default position that Adam and Eve looked like us, right? But young Earth creationists are constantly telling me that all organisms on Earth have devolved, right? They're very different than they were originally created. They've lost features. Like they're always talking about how this character is lost, right? Organisms are becoming, a complex canine has become a chihuahua, which has lost a bunch of information. And so if everything's always losing information, what about the loss of tails? Right? Why couldn't Adam and Eve have had tails? After all, they were created not having to wear any clothes. I don't need to the complications of having a tail and wearing pants, that wasn't a problem. Or did God anticipate that humans were going to wear pants someday, jeans someday, and tails would be like a, kind of a cumbersome thing. And so anticipated that. And so made us so we wouldn't have tails as a result of that. But you could always turn that around and say, like, nope, they originally had tails, and then God anticipated that the fall was going to happen. They're going to have mutations. They're going to lose their tails anyway. So yeah, we can wear pants then. It's like, so were you there? I just don't think we should make, I don't think that Ken Ham and others should make confident proclamations of exactly what something is when we don't really know. Right? Was he there? Was he there? He didn't see it. He should admit that it's possible. I'm fine saying it's highly unlikely. And in fact, I think Ken Ham, sorry, I just keep thinking of new things. So I'm just going to keep going. I think Ken Ham has got a little problem on his hands, because if he were to say that they don't have tails because they've lost their tail, right? It's going to be hard for him to say that, because that's going to look like an evolutionary explanation. Because he's going to be confronted with the fact that other primates also don't have tails and they have the exact same mutations. So it looks like common descent. So he's going to want to say they don't have tail. I'm just saying he doesn't have any evidence, right? All he has is like, I don't have any evidence otherwise that they did have tails, except that I'm kind of providing him with some evidence that they might have had tails. Because I'm saying that we all have the ability to make a tail. It's in our genomes, right? You can think of it as it's turned off. So then why is it there to begin with? Why is it there to begin with? I think the proper response to that, I'm going to help Ken Ham out here. I think the real response to that would be TBXT is necessary for a number of different functions. And so the fact that some organisms use it to make tails is just another one of the many functions that it has. And in human beings, it was never meant to have that particular function. But with the discovery of this 300 base pair extra piece of DNA, it's as if we're discovering that's what God added to a human being to change the function of this gene for humans compared to the functions that it has in other animals. Did human beings have tails? Interesting thing to think about. Let me know what you think. Am I just totally off my rocker here? Do you think it's possible that the original human beings had tails? I mean, I know that if you're a evolutionary biologist, or you think that humans have a common ancestry with other primates, then the obvious answer is no. Because this data provides a strong rationale for having confidence that, no, human beings have never had tails, because they share a common ancestor with chimpanzees and gorillas, and they both don't have tails. And it wasn't until you go back farther in the clade that you get to a point where there is a tail. And so that loss happened well before the origin of human beings, and therefore they would have just shared this information that they got from their ancestors, which is, you don't remember how to make a tail. Yeah, you got the basics of how to make a tail in you, but you've had changes that have occurred to your genome that make it impossible for you now to make that full-length tail as an adult. All right, so the evolutionary biologist actually has kind of a simple explanation for a simple answer to the question of, did Adam and Eve have tails, or did original humans have tails? The answer is no, and with high confidence. I'm just saying that Ken Ham can't have that same confidence. Where does he put his confidence? He can't say, put the confidence in Scripture. I can't put the authority of Scripture is my confidence that Adam and Eve didn't have tails. I'm sorry, the Scriptures don't say that Adam and Eve didn't have tails. Besides that, Scriptures record a lot of funky organisms that have some really wild features, right? Certainly not beyond the realm of possibility. All right, I feel the need to draw this to a close. I don't know what last words to say, so I'll say what I usually say, which is thanks so much for listening. If you're still here after what's going to be probably an hour even after I edit this, then more power to you. I hope something about this was stimulating. You learned something about molecular genetics. You learned a new way to think about Adam and Eve. and you learned a new question to ask Ken Ham the next time you meet him. Did his ancestors have tails? See what his answer is. I'd love to know. All right, we'll talk to you later. Thanks for hanging out here. Bye-bye.